Hey, so everyone can hear me, right? Just want to confirm. So, hello, bonjour, tout le monde. Assalamu alaikum wa rahmatullahi wa barakatuh. For those that don't know, I am grateful to be able to TA this course a second time with now 2 practicals on deck. So I'm TAing practical 2 and 8. And so for those that don't know, it's a pleasure to be able to introduce myself to you. My name is Dr. Mohammed Ibrahim Saeed, and I'm a graduate student, or actually a graduate of graduate studies here at UTSC. I studied cell and systems biology, and if there's any questions with regards to research, academia, and anything of the sort, just let me know at any time. I haven't written down my email yet, but if you have time to do so. How many of you have seen my purpose announcement? Please show a raise of hand. Can you keep it raised? This is fine. And I want you to reflect upon those 8 reminders. Now I'm going to look. How many of you have actually seen my announcement? So about half, maybe 2/3 of you. So I'm just going to share a few points, and I would recommend that you bear this in mind, because throughout your duration in this lab, punctuality, proficiency, and discipline are all keys. And what I mean by this is that be able to understand the concepts, the content that you're learning here in this lab of microbiology, but also more broadly, you're able to integrate your knowledge from lab and lecture material to novel situations inside and outside of your exams. So bear that in mind. It's not as simple as, understanding the concepts of a microscope, but being able to explore how you're able to, study different organisms, specimens, and even eventually cells, if you pursue graduate studies here in cell and systems biology. And just to confirm, everyone can hear me, right? Yeah, everyone can hear me? So just bear in mind as well that you've come in. I'm just going to do some very basic things to help elucidate this for everyone. You come in, take it off the you know, I like that you place your ability to find habits. And you watch again, right? You're doing now. This soap has all of this. And then if at any moment, let's say you touch a stain or like you have the stain or real stain on your hand, what you can actually do is while your hands are dry, make use of this lotion to wipe your hands dry and actually remove on any stains that you may have on your any questions? Any big questions?
Yes, so we gotta stay on our hands.
No, yeah. If you have a stain on your hand, use it.
Okay, and then we wash it.
First, yeah, exactly. So first you can actually use this. Because likely it's possible that you get crystal violet, for example, on your hands. There are different dyes around your benches. So just be mindful that if you run into the case before leaving the lab that your hands are color coded, and when you do just make use of this, essentially wipe it across your hands and then wash your hands after to maximize cleaning. And for whatever reason, bear in mind we use buttons for this lab. So for whatever reason your hair is not or if you have latex gloves on and you actually catch on fire, you can actually move the shower and put it on. I'm just preparing you in case of anything. But apart from that, in that first aid, you're here. And I would just say bear in mind the very important safety protocol when you're here in the lab, right? Because as you prep yourself for graduate studies or research or medicine, or even if you start your own lab, you want to be able to bear in mind certain safety protocols for yourself or everyone else. In terms of other things, any other questions? Make sure everyone is trying to the desk and interact with you. So other things to bear in mind that you're wearing safety glasses or goggles, you're having your lab coat nicely tightened in, and once again making sure that your shoes aren't exposed, so such that if there's any contaminants or explosives, they don't seep through water. And in terms of other things here, in your benches, you have a variety of items at your disposal or around you. I'm going to just show it on either side, but we're going to be replicating this very shortly. This is 95% ethanol that you're rarely going to use for at least 6 weeks. These are your bacterial strains that you could be using for the exercises of this week. This is 75% ethanol. This is water. This is your staining, let's just call it staining tray, where essentially you're going to be adding in across your slides different stains. So you bear in mind that you're using stains on this tray, silver tray that you see here. Don't do it on directly on the bench, because you want to be able to make use of spraying or staining your bacterial smears, then dumping them into the Wescadine, that light yellow looking beaker. So just bear that in mind. All liquid waste goes inside those beakers. And anything else like the case of trim white and bibless paper may go into the garbage bins, which is, I believe, surrounding the lab today. In terms of stains that you see here, you can see them all. There's actually more as well. 4 are placed, I believe, on each side of 2 in each bench, but there are a list of them. Saffron, crystal violet, and there should be 2 more on your screen. Yeah, sorry. Methylene blue and Nigerson. So just bear in mind that you have, anyone else doesn't see it, you'll be using on the white tray, the purple one, crystal violet, and the red one, saffron today. And then in this red tray, it's those 2. Methylene blue and then Nigerson. And then to prepare that in place, pretty much that's the setup. They have a Bunsen burner. Bear in mind that when you're starting on the gas, and I'll let you know when it's okay to do so, but when you begin, bear in mind that you keep yourself at a distance. Don't go too close because the flame may actually come right in front of you. You don't want to get any burns. Just bear that in mind. Just for display, it'll be something like that. then thereafter, we'll continue with the lab. Just always remember, as we'll discuss shortly, that there's a way of how essentially there's a level in which how strong that thing should be, because you don't want to make it too strong or too weak. And so just bear that in mind, everyone. Any questions in the meantime? Any other questions, thoughts? So let's see if there's anything else. Aseptic technique. Before we even begin, I should make mention of this now. Make sure that you stain, you spread 75% ethanol or 70% ethanol across your bench. So pretty much close your lab once we begin formal experiments, and then just spray around with 75% or 70% ethanol across your bench. Wipe it down with some tissues, and then you can begin. That way, we promote aseptic technique throughout the duration of this practical. And I mentioned most of this, if I'm not mistaken. Do not eat or drink. If you need a cell phone for recording anything, just let me know in advance. Yeah, at the beginning and end of the lab, make sure to wash your bench with ethanol. And let me just see anything else. Okay, and then disposing. Slides and glass pipettes can be placed in a Wascadine, but in terms of other materials, especially the ones that are found here, including the depression slides, the sample slides, and the ocular micrometer, bear in mind all of this has to be returned back. Please do not throw it into any of the bins. Just let me know if you have any questions, but we want to make sure that we can keep them for future use. And then before you leave the lab, try to wash your hands. I think hopefully everyone is able to understand the lab manual in terms of what exercises they need to complete. And I just want to also, sorry, any questions? No questions, no questions, okay. So I wanted to say as well that, so we have the exercises that we're aware of today to complete. Bear in mind that if you haven't, once again, drawn out the microscope, that you do so today, as well as there are discussion questions that if you haven't seen on my Quirkus announcement, you want to prep yourself of answering them in advance of the lab. I'm marking not only your lab notebook, but your performance, your attendance, and hopefully your participation in the lab. So bear that in mind, everyone. I want to make sure you all ace this lab and do your best to perfect it, but you just have to put that effort early in advance. And so with a light microscope, this is pretty much a setup of what I was referring to earlier. And so really, these are key components for you to understand, not only for your lab exam, but much before then. In fact, for the exercises that we do today, in the upcoming weeks, you want to be able to know how you're able to adjust or troubleshoot, if needed, the microscope. Now here's like a participation question for you all. What's immersion oil? And maybe why do we use it? And can you remind me your names? I just want to like regurgitate it for myself. It would be really appreciated. Rahma, okay. That's okay. That's okay.
This is something with high resolution. It's not going to work because the more denser the oil is, the more the distance between or the minimum distance required for resolution is lower. So we get a clearer image.
That's
so
great.
Maybe that's why we're using a denser material like oil instead of just air.
That's a great response. Thank you so much. And I believe there was another. So at the fine magnification that you're using, which in our case would be 100x, yes, the idea is that you increase resolution, which is pretty much by increasing the refraction through the index of 1.5 for oil. So yes, technically you're not increasing magnification, but you're increasing resolution. So that sometimes is is a concept that people may forget. But you're maintaining that same magnification at 100x. And then once you add that immersion oil, that actually helps maximize the amount of light that goes through your specimen, such that you're able to see it through your objective lens. Any questions on that? And so, and we'll be able to, hopefully you'll be able to see or understand how you're actually adding the immersion oil. Essentially, once you have your microscopes, which I'll just show you, is on both sides from these cabinet doors from here. You essentially open them up. You'll grab your own microscope shortly. But when you're in the mode of actually adding an immersion oil, I'll let you know what you do. Essentially, you're going to be in between two objective lens, 40 and 100x. And then thereafter, at that moment, you add an immersion oil, and then you slide in your 100x objective lens. So that way you don't have to worry about adjusting the slide or the stage itself in its position. I'll show things in due time, but I just want to make it or help you guys understand the material early on as well. So I think let's actually go straight ahead. We'll pause here and grab the microscopes. So I said them earlier that you need them here. So you can actually grab from this side, microscopes here. Make sure you grab the ones on the top or the ones that are light microscopes, like compound one here. There's other ones that are different. So bear in mind you're grabbing this. I'm just going to show it for display here. But you can grab them. You can make note of the one that you use. And if you find them to be any difficulty, just let me know at any time. You can take them as you wish now. Also, thing is, when you guys are grabbing them, so if there's anyone that has a bigger, and you can take other ones. Then you have the wires that are here as well. It has a flickering light. Are there any rules that you see? Oh, okay. There are. We can. Does everyone have a functional light microscope? No? If you need one, then there are extras here. I just want to make sure everyone has the right one that they need. And also, just in case if I forgot anyone in attendance, is Noel here? There's a person named Noel here. Okay. Everyone okay? So now that we have the light microscope, what do you think is next? Well, actually, I'll let you guys set up. I don't want to delay anyone in getting ready to prep up. But in the meantime, make sure that you have a wire, a plug to connect to the outlet and with your microscope. Okay, so when you're ready, let me know. Bear in mind as well, if you're using a phone, you have to let me know in advance, okay? I don't want anyone making use of a phone without letting me know. Otherwise, if Dr. Burn comes, I don't want you guys using my phone. Bear that in mind. Now, once you're ready, as you probably saw from the lab manual, that you want to adjust your interpupillary distance between the objective lens so that you're able to focus on high mag on your slide. We'll be using slides shortly, but in the meantime, be familiarize yourself with the microscope you're using. Try to pinpoint for yourself where the nose piece is, where the diaphragm is, where the fine adjustment knob is. So that way you're able to, with ease, manipulate or make use of your objective at the best, or you make use of your microscope in the most efficient of ways. So now in terms of specimens, we're going to be starting shortly, but I want to make sure everyone has an idea as to. So, yes. And in terms of discussion questions, I mentioned it earlier, but make sure you try to answer them in the lab notebook, as well as those that are from your lab manual. So that way you'll be able to understand how you would be able to best excel in the course and also complete your lab performance throughout the duration of our practical together. So we have, now you have your microscope, right? Now I'll prepare a slide of bacterium. We do have a bunch here, so I'm going to show you, but I'm just trying to see if we have the one that you need. We're going to be actually preparing slides as well today. So to make it easier, we have 24 of us here. Okay, so what we can do is make use of these slides here today. I'm going to place them on. Actually, if you take each person grabs one slide and then brings it afterwards, just so that way they're able to resolve their magnification. So you can just grab the slides and we'll be able to go forward. And this is going to help us for exercise 6, which is pretty much the last exercise for today, because you'll be able to see the flagellar stain of different microbiome. And so the idea is that you'll be able to deduce, okay, what, and I would highly recommend everyone to draw, a figure in your notebook of what that microorganism is by their flagellar arrangement. So, and hopefully you understand what I'm referring to, according to exercise 6. Everyone's grabbed the slide. Yeah. Okay. So now what's next? I want to see if you guys are understanding what's next for today's practical. So you're gonna look through the microscope, have a glimpse of your bacterium. Hopefully you're able to adjust, and as you go up towards increasing from the course adjustment knob, your stage, you'll be able to see on a higher objective your microorganism. I forgot to mention, but I think some of you may have so I think maybe some of you may not know this. Can I have everyone's attention? For those that haven't actually adjusted yet the microscope in the very beginning, you have here's the following. Log on, so you're able to turn around the microscope. And that way, you'll be able to actually have your objective aligned with your slide. So just bear that in mind, because I know some you maybe have it converted. Let's see. So we're going to be studying make sure indeed of the pattern. Are you able to focus on your slide? So what I would recommend is if you're starting at 10x, try to first get an idea as to where you're oriented the light source to pass through your specimen. So I'm going to just talk you through it. So as a top, any questions on it? To actually zoom in and see their organism. If you're using the ones that are also familiar with slides, you can actually make use of them to draw them out. Which you'll likely do down the road. But so that way you're able to deduce things coming up shortly. In the meantime, though, let's just see. We could begin with the ocular micrometer if everyone's okay. If you at timing, if you feel like I'm rushing you, just let me know. I could wait for a few minutes. But we could also move forward and go straight to the ocular micrometer. Just want to confirm for everyone, are you able to now see your bacterium? Can everyone see anything in the slides? Anyone having any trouble? No? Okay. Everyone else okay in terms of seeing things through the microscope? Is anyone having problems with the microscope? Such that it's visible to see anything? So, yeah. So in the meantime, I think we just anyone else having problems, by the way, with the microscopes? Is everyone able to see through their slides? Everyone's able to see bacteria? That's the main important point for now. And then afterwards, we'll be able to use immersion oil and then forward from there. Before we move though, I'm going to actually separate you into blocks. So think of it like this. Each set of 4 people, 3 to 4 people on each side will be considered a block. Meaning, I'm just going to separate. It means that you're all one group, you're all one group, you're all one group, and you're all one group, you're all one group, all one group. I'm saying this now because there are times where you'll likely be working on group of 4, sometimes at here, and often individually. Making sure you're able to understand how you're going to calibrate your optimal micrometer. So what I've done now is separated the optimal micrometers for you all. Focus and complete exercise part 2, exercise 2. So what you're going to need to do is using one of the microscopes that you see I trust with resolution to be able to take one of them per block or per group to be able to now determine the following. Sorry, let me clarify. Not for this one, from the one that's in your lab manual. In your lab manual, you have one that's similar, but it's just left blank, right? And so I want you to be able to use at least, for example, at high and oil, so 40x and 100x. I want you to be able to determine the different values, but also determine the calibration factor. So that's what you're going to do for most of today, for at least the next 20 to 30 minutes. Any questions on that? No questions? Okay. So now you can just grab, in the meantime, I would recommend everyone who's ready to grab per block one of those purple or violet boxes for your block. Okay. Yeah. Yes. So once you're done with the slides that you used for analysis and visualization, you can place them back to where you found them. Make sure you do not dispose of any prepared slides. Thank you, everyone. What?
Oh, okay, okay.
I was thinking in terms of using the optimal micrometer.
All four.
If you could do all four, pretty much as a group of four, it should be doable. If you could do that, that would be great. Great.
Other than that. You know, you get on the benches. You be carrying them everywhere. Then your glasses off, get under your. Oh, okay. We can always do what I do. Do that.
Are we able to know the longer to run things from like our
That's I'm starting to get out to
And if you need to do works, you should be
because they can always continue to do this while their smears are
drying. Yes. Okay.
and that's all.
As a group, you guys will be. So in the meantime, I think for timing, something that Dr. Brent noted was that for timing, we could go straight to the bacterial smear preparation. Don't worry, because generally speaking, these are the main two components, the smears and the ocular micrometer to focus on for today. So, I mean, we could probably be done in the next few minutes, the micrometer, but I would prefer that you begin now with the smears. I'm going to turn on the gas if it's not already on, and then you can begin in the next few minutes. What you could do now in the meantime is just have a look back as to what smears you're going to be doing. There are multiple smears, multiple stains for today. But in the meantime, just have a look back at either your manual or your lab notebook to familiarize yourself as we move forward with exercise 3-6. Any questions? Yes. So just so everyone's aware, in terms of the ocular micrometer, what we're going to do afterwards is at least be able to see with your left eyepiece the ruler that you're going to be using to overlap. I'm going to show on both sides. The start of your eyepiece micrometer is the stage micrometer. You have both of them, right? So what you're going to be doing is aligning them from the very beginning where the two zeros meet. Hopefully you see it on the other side as well. And so the idea is that you're going to be using that as a framework to determine the different stage divisions, but also how much they are in size in microns or micrometers. So just bear that in mind when you're using these values or deducing the calibration factor at the very end. But essentially, you have all that you need. Just make use of your micrometer accordingly. But I know that the screeners are going to take some time, so we're going to go forward with them in a few minutes. Hopefully, though, for everyone that I've spoken to thus far, has fixed the resolution for the microscope. Otherwise, we can begin and then carry on in situ or like in junction with the ocular micrometer calibration. So hopefully that's clear for everyone.
I'd like the two lines out.
Here's when you write your notebook. Also, extra information. Relevant questions. Yeah, we're being individual. You have to do your own notes individually, so you better practice it yourself. Otherwise, you're not really hitting your assignment. But make yourself a number of smears, follow the directions, air dry them while they're air drying. You can continue with your ocular micrometer. Once they're air dried, you're staining, and you can look at them. That's when you're also starting your makeup draw.
Yep. So So actually, how about I'll make it each. So the other one,
But this one's supposed to have two. Remember, your goal when you make a smear, it should want not put a lot of bacteria on the slide. That's usually your go-to, because you think you're small and need lots. So when it air dries, if you see a very thick smear on your slide, you're gonna have too many bacteria. So that's why you're gonna practice this, because you understand that. Okay? So you should start making those smears. Smear, I can make it smear.
Number of things. Oh, bigger one, which
Okay, it shows them how to accept with bacteria workers on.
no, I haven't passed it yet. I was gonna show it to them very shortly.
Okay, because we need, because this kind of, this class can, it's gonna take 3 minutes for the slide to draw.
Yeah.
Okay, so how would you do that on board?
Okay.
So this one is the.
Yeah. The large one. Perfect. So everyone, I just want to make sure we're on track. And I do want to apologize if you found that it was a bit more independent with regards to the instructions, but I'll provide some demos, at least for the smears, of what you need to do. So in case you don't know, there's a few things that you have to bear in mind when you're doing aseptic technique. I've mentioned this earlier about, and make sure everyone has done this, hopefully, to spread or spray 70% ethanol on your bench. Make sure that your bench is clean before you make use of. Now, as we move further, I'll be able to show you a demo. Now, this is for, don't worry, I would say too much for now. If you haven't done so already, exercise 2, we could go straight to exercise 3 and then come back to exercise 2. The aim pretty much for today is to do, okay, so it's the simple staining and other stains as well, but moving back to aseptic technique, I want you to be able to notice a few things. Can I have everyone come to this side if it's okay? So I'm just going to show a demonstration. Is it okay if I get so, everyone just have a close look. So before we move further, there's a few things that you have to bear in mind. One is the following. You can see here's a few things. So starting off, you have flame that's on. I press or turn on the gas, but what you can see is it's quite high. I would actually prefer if you could go a bit lower. Make sure that it's not too high for you. Reason being because you don't want to get any clothes catching on fire from your ear. So bear that in mind. You can go from clockwise to counterclockwise, and just bear in mind that you don't leave it unattended. Very, very important for safety protocol. And then once you make use of, you can make use of your loop, roughly decoating to actually flame it. Make sure that it's turned red, so that way you kill any bacteria or on these aseptic taken care of them. And as you open your tube, any tube that you open or close, make sure that as someone else or yourself ideally makes use of the inoculum. I'm just going to show for demonstration that it turns red. You see, you wait for a bit of time, so that way as soon as you get into your bacterial liquid, very, very small at the very edge of the ring. But then once you're done, what she has already done is you place it on a droplet of water. And as you know, bear in mind, I'm using Bunsen burner, so I want to make sure that once I'm done making use of the inoculum loop, once I've dabbed in the given amount onto the slide that's labeled with the bacterium, once I'm done that, I just once again essentially flame off the inoculating loop, and then repeat that process, making sure that no bacteria comes in, right? So that way you can make sure that you're not contaminating any of the organisms or your bench. And then if you, for whatever reason, feel unsure, or like myself, I've done it more than once, you can actually do it again. Then you call it back here, and then your tube will be placed back where it belongs. Just want to make sure if anyone's confused with what bacteria we're using, look back at exercise 3 to see the stains. You're using simple stains, right? But bear in mind the organisms that you're going to label. You could use SEP. Normally I like to be very descriptive, but because of the amount of space you have on your slide, you could just write it on the opaque space, say, like for the left-hand side, S.EPI, or S. Staphylococcus epidermidis. Any questions on that note? Once you're done, you actually take this to the dryer, or sorry, the slide heater. So if you have a look, I'll just show here. Once you're done with your smear, you pretty much come here. It's already warm, and so you let it warm your slide where the smear is going to dry up. Any vapor, any water, such that your smear is now going to remain intact onto your slide. Afterwards, that's where you're going to use the slide tray. We'll talk about that after. And in the meantime, any questions? Everyone understands how to do aseptic technique. Essentially, bear in mind that you're not too close to the Bunsen burner. But whenever you're opening or using any bacterium, that you open the tube, swipe with a Bunsen burner before you close and when you open. And when you're using the inoculating loop, you do the same before and after. Also, once again, just make sure everyone that when you're using ethanol, you're not too close to the Bunsen burner if it's already on. Before it's on, though, just make sure that you always spray before and after. There's 3 things that you're doing before and after. Your tube, flame once and twice, your Bunsen burner, and the ethanol that you're using at the beginning and end of every batch. Bear that in mind, everyone, especially when it comes to using bacterium, because you want to make sure that the smears that you see and the slides and images of cells that you see actually represent the bacteria you have here and not from the airborne environment. With that, you can continue. This is more of independent work, so just make sure that you're able to complete this, and then we could go back to the ocular micrometer in the minute afterwards. Thank you, everyone.
How much water did you drop in this?
1 to 2 drops.
With that bottle?
Yeah. I can help you. For the liquid, yes, only for the solid. For the liquid cultures, everyone, I just want to make sure you understand this. For the liquid cultures, make sure that you already have liquid presented on your smear, so you don't have to actually add in more water. For the solid agar, where you have it finely tuned, not as a liquid, that's where you're actually adding water. To dilute the smear. The liquid ones should be I think yeah, Yeah. So this thing is, however, I want to use to everyone okay? So I just want to make sure everyone's not burning themselves. Make sure that when you're sitting on the plane, you're not too close. Bear that in mind, and nor should the ethanol be. A few other things that were asked about liquid agar, the liquid culture versus the solid culture. Solid culture, you're adding in drops of water to your slide in advance. Bear that in mind because that will help make you dilute and place accordingly or suspend your bacteria in your cylinder. Also, in terms of the stains, for simple stains, you are making 3 slides. If you wish to take the slides, they are here. For those I don't know, the slides are here for the ones that you're preparing for your smears. For exercise 3, you have slides that are here that you're going to label. If you need more, and in fact, you're going to be using only 3 for today, so bear that in mind. You could grab them all in advance, so that way you could do as many stages as possible, and then we can come back to the ocular collaboration. 1 moment. 1 moment. I would recommend You could not only air dry like on your bench, but you could use the slide warmer or heater here. Make use of it so that way you can heat up your slides in and in due time you can go back to the ocular micrometer calibration. Everyone, make sure that you're being mindful of your experience. That it doesn't touch into the tubes of the Bunsen burner because you don't want to have any contamination. Once again, if you're not using the flare of the Bunsen burner, make sure that it's off. How are we doing in terms of staining? In terms of staining, you only need to add 3 or 4 drops. Not even 1 drop is enough. Actually, one. Everyone, when you're making use of the loop and the tube, bear in mind you're using both hands at random. Try not to put anything down the lid or the loop because that way you reduce risk of repeating the steps for sterilization. So imagine I'm opening it up like this. Use the loop. I'll leave the loop and the lid, and then I'll go back.
bottom right, the units record the size of the shape. And the range for that.
How's everyone doing for their smears? Oh, yes.
Now, you have yours is better than mine. Okay. Oh, yeah, we got yours. Yeah, I know. Yeah, on the table. Mine is from. Mine's from the bottom. Yes. Oh. Yeah, do you think approximately how long?
Fine,
Okay,
even maybe that, how long have you had yours? How long have you had yours?
oh, like about a minute now.
That's okay. Make sure when you place your slide on the warmer that it's not, that it is labeled, right? Bear that in mind so that way when you come back, you know which one's yours. About 3 to 5 minutes. 5 minutes should be enough time. No problem. And if you can as well, try to know what you're doing as methods for your lab notebook. That'll make it easier for you to come back to bring back to the example.
What I'm suggesting before the. I'll say, I like to see it.
How are we doing, everyone? How are we for timing? Do you think in the next 15 minutes we could be done our smears? Sorry, not our smears, our stains. Yeah? For context, the staining shouldn't take too long. Usually it's about 5 minutes, but giving you 15 minutes, hopefully that will help you complete the remainder of staining. I'm going to just skip to some other slides. This is what you have here for discussion. So hopefully everyone's paying attention because this relates to exactly some of the questions that have been asked in terms of cell morphology and arrangement, right? If you're going to different objectives. And I know that's actually why I didn't want to skip the calibration component, because determining the size is what you're going to need in advance. But the idea is that when you're using, or actually when you're trying to determine the size, I would recommend noting the objective that you're at and the number of increments that you see from your ocular eyepiece. So bear that in mind. Pretty much you're going to see something like this for, let's say, panel A. And you want to be able to determine off the microscope what the exact size is, or roughly, approximately in microns. So that's what we're going to do. It combines exercise 2 and 3 with the simple staining that you're doing now. And then similar to this, you'll be able to use your lab manual to derive the different cultures that you would expect for A and B. And this should be understood because one of the noticeable differences is their shape. So bear that in mind, and also color when you do different types of staining tests. So that's something to know to determine, once again, the cell dimensions. I mentioned exercise 2. Negative stain, I do want you to at least see what we have from the samples here. The hanging drop method we're going to be doing shortly. But that's what I want you to do before we end off today's class, following the ocular calibration. The flagellar stain is relatively easy to determine. We did this earlier, but you can have a look back at it afterwards. And then this, I believe, yes, will be done in week 2. But if there's any questions upon it, just let me know. If there's questions also, just let me know. The goal is, in the next, I would say 10 minutes, we should be done our simple staining for each person in the classroom. Make sure that you've labeled your slide, and then we'll be able to place them into a location for reuse up in next week, hopefully. And then after that, after 10 minutes, we're going to be going to look at the acidic stain. Or even hanging drop for 15 minutes. I want you to be able to do your own hanging drop, which will lead us to about 11:15 or 11:20. And then afterwards, we'll go back to the calibration for 20 minutes. That'll be 11:40. And the rest for any outstanding material, including the flagellar stain, which could be done in about 5 minutes. So we'll be done hopefully 11:45, but I need you to maintain a level of pace and cooperation accordingly. Thank you, everyone. Make sure, please, that you're not taking too many slides as well, because we want to make use of them later on. And when you reach the hanging drop, let me know. Thank you. There's this paper right there. Yes.
I don't know what I'm looking for.
One moment. It's a bit. So, yeah, this could be possible. I would recommend, if you want to find where you're going to look for visualization, try to look right there where the fainted areas. Yeah, it's a bit difficult, but I would say probably right where it's projecting out. Yeah.
And then after I viewed it, I can do the negative.
The negative sign. Yes. No problem. But it's very simple, just like what we've done before. Normalcy will be used for the United States, is violent, definitely bruised, and harmonious is used for simple states. In order to do violent. So for those that are wondering about oil pollution, what I said earlier is the following. You have 40x and 100x objective lens on two opposite sides, i.e. your slide is going to be just exposed to the light. Thereafter, you add in, essentially as you rotate the objective lens, you add in that immersion oil and then you slide it into 100x. If there's difficulty on that, it's not too difficult, but if you have problems, just let me know. But if you need that for calibration, you may not really need it, but depending on your bacterium, you may need to see further for your resolution. For us to done, you can put across slides there, just be mindful for depression slides. You place them back, you wash them, and you place them back, back to the movement. Just gonna answer the question. No questions? Yeah, it's okay. But before we do it, how's everyone doing so far?
So what do I think data? Yeah, okay. Okay. I think that's. Yeah. Thank you. Lights. Go up.
Okay, before I go there, how's everyone doing so far? I'm trying to get on the edge. Updates. How's everyone doing so far? Can I ask, by the way, I see a flame that's too high. Group from the very end, yeah. Can you tone down the flame? So a few important things for everyone. In terms of timing, it's 11:18. And so really by now we should be done our stains. In fact, now we should be moving to the hanging drop. And I want to make sure everyone has time to do this. Now, in terms of other things that maybe you've noticed by now, is the importance of staining the right amount and to make the right level of smears. Is everyone paying attention? And it's very important that when you're making your smears that you add in just enough such that you're not overstepping with too much in that way that there would be so many layers on top of each other, like limestone, like if you think about like rocks over one another. And it's not too limited such that you won't be able to find anything from your specimen. So bear that in mind. That's number one as one of the limiting factors that you can discuss for your post-lab, but also thinking about more broadly your protocol for today. Second point is your staining technique. How much stain are you adding? For how long? And even in terms of your smear, are you adding too much water or too little water? Because if you add too much, it's going to dilute. If you add too little with the stain, it's going to coagulate, meaning that you're going to have so many colonies grouped together. It'd be very hard for you to determine and visualize isolated colonies. So bear that in mind, two important features for smearing for today. And for next week, this will help prepare you more for upcoming staining techniques. So don't worry. Really, the aim is to be able to understand how you're using your microscope, how to be able to, what we're going to show is determine calibration for your sizes, and being able to visualize and smear accordingly / stain your bacterium. So bear that in mind, those three key components for today. I do want to make sure though that we're able to hand off with the hanging drop. So is everyone ready to go there by now? Yeah? Everyone's okay? Are we able to? I apologize if it's quite tense for you, but hopefully, like as things go on and as you're able to familiarize yourself following this week's lab, you'll be able to understand what you need to do next. Going forward with the depression slides, I see some of them already open. So just bear in mind that when you're done, you bring them back. Now, in terms of the depression slides themselves, there's a few things that you bear in mind. One is that you have small square slide, essentially covers or cover slides that you're going to be adding Vaseline to the edges of. So here's what you're doing. Essentially, you're replicating what you've done for staining, except rather than doing the staining procedure, you're getting the liquid. Sorry, you're getting the liquid culture. You're adding in a few drops, 1 or 2, max 3 drops of your bacteria. Then maybe I should show it on the slides. They may show it here, but the idea is like this pretty much. You have your cover slip, you add a drop of bacteria right here. Then on the side of your depression slide, the one that has the depression where you see and feel that there's an edge. Did anyone take a bunch of depression slides with them? Okay, a lot of them they're not perfect. Thank you. So these ones, yes. So the idea is that using Vaseline, right, there should be on each side or each block of benches, you should be able to add Vaseline, 4 drops on the side where that depression is held. So for me, it's actually underneath. So bear that in mind. That you add in 4 drops on each edge of Vaseline or petroleum jelly. And then on that note, the side that has your bacteria, you essentially invert your depression slide on your bench, which should also, by the way, be cleaned with 70% ethanol. You just simply add in and you let the 2 surfaces touch essentially, and you quickly invert. And then you should be able to see with ease and as efficiently as possible your bacteria to determine their motion. So we learned in the lab manual that there's two types of motion. Browninian, which is like more of like vibrating, non-motility essentially. And then you have true motility where you see bacteria like E. coli swimming around. So bear that in mind. When you're done with the hanging drop, then what you do is normally see it from the edge. But when you're done, you clean it up with 70% ethanol, and then you bring it back to here cleaned up. Make sure that you don't throw the cover slip or the slides away, but you make use of retrieving them back here. And that should be it for slides and smearing for today. At the very end, though, I would say by 11:45, actually 11:40, we're going to be coming back to do the calibration, and that's where we're going to call it a day. So hopefully everyone should be done staying in, but now we're going to move straight to the hang and drop. Any questions? Any questions? Any questions? Okay. Yes. Okay. So with that in mind, for anyone who is ready, you could come to this side of the bench to be able to grab a cover slip and a hanging drop piece for your group. Or sorry, for pairs, you could technically do it as groups as well, but try to do it as pairs. Yeah. Yes. I would say do the hanging drop now. Okay. If for the negative scene, I think for now it's okay. If you have none, it's okay. Sorry. Do you want to come this way?
I just need the stuff. Yes.
Perfect. These divisions, we're going to talk about them, but essentially they're the ones that you feel in your ocular micrometer. Yeah, like how many you see. That would be 0. If the increments are right. And then when the value itself of each micro division is similar to then you simply multiply and then you actually compare that with the own and then you divide the two, and then that's when you get calibration. Oh, okay. I have as a suggestion for you all. If anyone wants to keep their slides, you can. Just let me know if you wish to. But in the meantime, I would make sure that you're able to perform the rest of this the lab with proficiency.
I don't think there was. Maybe I'm.
guys need help for the hanging drop, what I would recommend is for the hanging drop, bear in mind that you're not going to really need the Bunsen burner, so hopefully everyone could shut it off in about a minute. I do want to make sure that in the last 10 minutes or in the next 10 minutes that we're able to proceed with the hanging drop, which should only take 5 minutes. But I want to make sure that you guys as pairs are able to do this. And if there's any outstanding measurements that you need to be done, then make sure that you have a separate microscope that you can make use of for determining the motility of your bacteria. So bear that in mind, everyone. And then we could come back in 10 minutes to discuss further the calibration. No, not.
You said one or two?
But you could use, I didn't see two before. It's my first time. Oh, so I normally see one, that's why. If you see two, that's also really good because I could save you time for doing multiple. For one for E. coli, and I think there's another. If there isn't, then it's okay. Just use it. You can feel it. You can actually, and then you'll be able to determine it. When you're the pressing side, which side is the one that has that pumping edge? You just touch it directly in the other side, so you're going to over when you make it here. How's everyone doing? Bear in mind that 11:40 we want to be done, or I'll say 11:41 to be done the hanging drop. So if there's any questions, subscribe to ask.
Yeah. Oh.
So everyone, if you wanted to know how you're going to determine on your microscope once you've done the depression, so once you've done the depression of your slide with your bacteria, by the way, there's a fire. So like, I want to make sure whoever's using the flame, you tone it down, please. Okay, so I just want to make sure that, and if you smell a lot of smoke or fumes, that's not a good sign. So just bear in mind if you're using flames, tone them down. But for your slide, sorry, the depression slide, if you wanted to see where motility is, it's a hanging drop, right? So imagine your drop is going to be going downwards. So the idea is that at the edge, where there's going to be only generally 1 or 2, 3 layers, you'll be able to see your E. coli and their motion. So try to look at the edge of your drop, not at the direct center, because you may see it will be very difficult actually to focus, which with multiple layers of slices, essentially optical slices in your microscope. So try to look at the edge for understanding edges like around here, around the periphery. So just to bear that in mind, the edges around which your Vaseline is surrounding. Any questions? We have about 5 minutes for the hanging drop. For everyone, it's important that we focus on continuing with the technique. Right now, I want you to be doing the hanging drop. Don't worry about the other stain, because it's important that we continue forward. And if we go back for next week, we could go back to the other stains if needed. Otherwise, I just want you to continue with the exercise. Go to the hanging drop, finish it in 5 minutes, and then we'll go to the calibration to end off or to complete the rest of today's practice. Bear in mind as well, I know I said turn off the flame earlier, but now if you're actually doing any stains or even actually smearing with the hanging drop, that's your time to turn on the flame again. Once again, make sure it's not too high as you start. Start off small and then you can increase accordingly afterward. Make sure that you're playing, you're not really loose the lid as well. It's as long as you remember that you need to do.
And it starts next week. Okay.
But I would say just make sure you're good to draw what it said from the simple state. If you can as well, write a figure legend or caption mentioning what bacteria you use. The not just like with the morphology, yes, but with the objective lens that you use. The magnification is very important as well. And any other features that you would want to include.
Please check the drag.
Yeah.
Yeah. After the lab.
There's one pre-lab, ones that are for this.
Like prior to the
听人没来，你了。
这个得是尽量少细菌，蛮好的。
Yeah. So those are
Okay. Okay.
no problem. I'll be back. Sorry, you had a question. Okay, so how's everyone doing? Are we nearly done the hanging drop? Have we been able to visualize E. coli and maybe at least get an idea as to, okay, if you haven't, actually, I want to have a raise of hands. How many of you have seen, have done the hanging drop? How many of you have done the hanging drop? Okay. How many of you have seen E. coli? Raise your hands. Or your bacteria, essentially. No? Okay. So the idea is that, and maybe to reiterate this one more time, everyone, please pay attention, by the way. It keeps things as efficient as possible to pay attention. The idea is that your bacteria are going to be more determinant of their motility around the periphery, meaning that you're going to try to look at the edges where there's that gap of air, essentially, where there's been that compression of your bacteria. So that way you'll be able to determine if it's Browninian, i.e. it's vibrating, essentially, or it's true motility. And that way you actually see bacteria moving in different directions with their flagellae. So bear that in mind. Now, I do want to conclude with some very important steps for everyone. I've mentioned this earlier in terms of determining, visualizing your bacteria for the simple staining. You want to be able to take this into your figure your images of your lab notebook. So you're going to be making figures in your lab notebook as your results for today. Your simple stain with a diagram of your field of view, the morphology, the size, the arrangement of your bacteria. These are things you want to include on top of the name of your species or your bacterial specimen that you did or you smeared for today. Bear in mind the other features that I've talked about, the post-lab discussion points, limitations that I've mentioned about the smearing quality, the staining quality, things that Dr. Brent also mentioned recently, but things that you need to consider from the lab notebook and from your experience from today. Now, I'm going to go back to, and also don't forget the lab questions. Very important that you respond to them from the slides based on what we've talked about today. And this is pretty much the last exercise I want us to complete to focus on the measurements of calibration. So if you haven't, if you don't have the ocular micrometer now, and if you haven't been able to, or your group hasn't completed yet the ocular micrometer, this is the time to do so. So what we're going to be doing is, oh, I think maybe you already have it. There is one remaining here, be a sign that you completed the calibration. But the idea is that we're going to return back. At least one microscope should be devoted for the ocular calibration. So discuss briefly for the next 30 seconds who in the lab is going to be from your block or group going to be using their microscope for the ocular micrometer. And then I'll discuss further on how you're going to be doing calculations. If you're already done, then that's a good sign. Continue with the hanging drop, or you can start cleaning up because that pretty much concludes the day. I would also recommend if you're done the calibration, then you can bring back the belongings, the hanging drop, glass slides, depression slides, anything that we've used thus far, including the ocular micrometer set, you can bring it to the middle bench. And if you have any remaining slides that you want to keep, you can also place them in the middle bench. Just let me know as you complete for the simple stain of today, that exercise. But moving forward, ocular micrometer, I'm just going to tell everyone how to do it, and bear with me because I'm going to just say it once. I really want to make sure you're paying attention, as this is a mandatory exercise. So you have to understand the following. You're using a microscope, right? And you have two different rulers essentially. One is on your eyepiece, your ocular or eyepiece micrometer, and the other is the stage micrometer, the one that you're actually using from the purple box in the middle bench. So the idea is you're going to be aligning the two. You're going to align the two of them at the 0, and that could be at any magnification. If you're stuck on time, just focus on high power at 40x, and then that could be converted afterwards to different mags. But the point is, you want to be able to align them and see the number of digits. So for example, on the eyepiece, on the stage micrometer, you have the number of divisions shown here. All of them, it's essentially arbitrary because the units are not shown on the slide. However, you know, based on what the divisions are shown here, that each small division is 10 microns, meaning that each set of 10 is 100 microns. Meaning if we were to do the math, if you go to 100 microns here to about 15.2 divisions, 15.2 divisions or units on the eyepiece, aka ocular units. So 15.2. The idea is the following. I'm going to write it down to make it easier. You have 15. So I'm going to do a setup. Just imagine this was your case. Imagine you're at 100x, 100 mag, and imagine that's 100 mag, and you have your eyepiece micrometer aligning at 15.2 OCU, which is ocular units, or OU, ocular units, very similar. Now, the stage micrometer is showing your numbers by numerical value. So this is important for calibration, and that's why we get this factor here. It's going to be aligning with 15.2, right? So I'm going to 15.2 where my mouse is, connecting that with the 10th stage division. Each large division, once again, is 100 microns, right? Because each small one is 10 microns. So we're going to say essentially, I'm going to just show proportionality, 100 microns. If you do the math, you're essentially doing 1 OU divided by 100 microns divided by 15.U. And that's essentially your calibration factor, which is going to be something we're going to say about 6.3 or 4 in calibration. What that means is that for every OU at this magnification, for every OU, that represents 6.4 microns. So hypothetically, this bacteria is 2 microns. Or sorry, 2 OU? It's 2 OU. So perfect. It's 2 OU. If it's 2 OU, then how long should that bacteria be in microns or micrometers? 12.8. Any other numbers? 12.8. Any other numbers? 12.8 is right. So great. I want you guys to think like this because this is how you're able to do the exact same thing for everything else on this table and everything else beyond this course. You're able to actually not only derive the values, right? Because imagine this is 5 power. I told you the stage division was 10. That's 100 microns, right here. And it's lowercase m, but just so you know. And then OU is what I said was 15. I don't know, it's like pretty much the same, 15.2 roughly. And then the calibration total value is 6.4. You just do very simple reiterations of this process. And the example table is here. But the point is you want to do the same with your values. So that way you're able to do this down the road with your own measurements. So the next 3 minutes or 3 minutes, let's say, I want you to be able to do this. Look at your microscope, even just at 1 magnification. If you can do more, the better. But do it at high, 40x, and just see the values, where they overlap, and how much, let's say at 10 digits or 100 microns, are you able to overlap with the eyepiece to derive the OCU. So the idea is that these values, the ones on the bottom ruler, they represent the first two sets. So very value now. And OU is the one that's on top. So OU is the one that you're reading from your eyepiece to monitor micrometer or ruler. And then the one that you've added in from the stage is the one that you're using from the bottom, and they represent these two divisions. Then when you derive the calculation, you pretty much represent what that value is, where they check. If it's 15.2, you add it here, and then you simply divide stage micrometer measurement by OU, and that's what your calibration factor is. And you just repeat that process for other magnifications whenever you can. If you can't, it's okay, but focus on at least doing one. If you can do two, that's even better. And then we can conclude the lab with cleaning up. Thank you. So just wanted to say, so well done also for the lab. I apologize if things are very hasty, but I want to make sure you're able to complete as much as you can throughout the duration of the lab. So bear that in mind from my announcements as well. Punctuality, very important. So just keep yourself organized. If you feel like you're being behind even 5 minutes into a given exercise, slow down and see how you can move forward. Microscopes, you can actually leave back on your desk because we're actually going to be using this this room for the next 3 weeks, and labs will also use the microscopes, so you can leave them as they are. Now, in terms of 2 last things, or maybe 3, when you're done with your stains, actually before this, you can take photos of your hanging drop. You don't really need the hanging drop just for photos. You can just determine if it's true or Browninian motility. In terms of other things, determine the size of your bacteria from the OU calibration that we've done today. That needs to be done and is part of your post-lab. So bear that in mind for your calculations. After this, then when it comes to Brownian motion, the slides, you bring them back, the Prussian slides, the cover slips. Bear in mind, those have to be brought back here. The ones that you, so the demo slides, the Prussian slides, bring them back. Anything else, any slides that you use for today, you can place them in the Wascadine. If there's anything liquid, you place also into Wascadine. But if there's tissues, disposable wipes, you can place them into the garbage. Apart from that, oh, don't forget to spray your benches, very important. And then wash your hands and have a great day. That's all for today. I just want to make sure. Can anyone explain on? Can anyone explain on? So rinse it. But you don't have to physically touch it. Essentially, when you're using you're cleaning up your tray, just place it into the Wascadine feature and then rinse it off with that cloth. So once you're done, please bring back the ocular micrometers, the stage micrometers right back here. We're gonna also be placing back the hanging drop depression slides back here. Make sure you place them in an organized fashion. Make sure as well that they're clean with ethanol. And then you can make way to cleaning up your bench. When you're done, let me know, and then we can proceed further. If you want to keep it, no, actually, you can place it right here. If you want to keep any of your slides, also let me know. Technically, you could dispose of them, but if you, and that way, in the upcoming week, you could practice for more stains. But if you want to keep them, you can also keep them. That's okay. Just let me know. And in fact, please place them close by in the middle bench. But we may not actually use them for upcoming weeks, or we may make use of other ones. Okay. So you want to put your yeah. And for E. coli, it is true motility. I want to let you know this, but you'll also see how in other modules, like other experiments that we'll be doing for the motility test. And so just bear that in mind. Sorry?
Observations for people.
I'm going to refer to it. So just once again, when you're cleaning up, make sure to wipe your bench. You can wash your hands as well. I just want to reiterate for post-lab, what are you supposed to do? Make sure you draw the figures of your simple stain. Once again, with a caption detailed of your magnification, the bacteria that you studied, the arrangement, morphology, etc. And then on that note, when it comes to your tables, the ones that we've done today for any exercise, when it comes to the calibration or when you're doing other images, make sure that you have them included for each exercise for what's to be completed. For the discussion, that's where you're referring to what your results are, what your observations compare to what you have expected. For example, I told you that E. coli has true motility, so compare that with what you expected. Or for the simple staining, see with what you've observed, what you could have done better. Could it be that your smear or your staining technique could improve, and see how? Make mention of these points because it's key for your discussion, but also for the critical thinking questions found in your lab manual and the discussing questions found in the lab slides. So bear that in mind, everyone. And I hope you enjoy the rest of your day. Bear in mind, we also touched about this earlier, the flagellar stain, and motility tests don't worry about, but answer questions like even for this, classifying the different types of flagellar arrangement in panels A to E, something very important for you all. Take care though, everyone. Have a great rest of your week and all the best. Technically, you can see them before, but I will also see you. No problem.
Just put this in the list.
Is it yours? You can throw it. Not to the first one. You're gonna need to clean it out. So make sure once again, depression slides are returned back. The covers are also returned. Ocular economies, the microscopes, also make sure you clean them from the oil that may have resided on the objective lens. Make sure that they're clean. Make sure that you're not letting anyone to come in and find any traces of one or things on your microscope. 2 minutes. Everyone cleaning up, make sure that you shut out your microscope from the back. And then in the discussion, and what they match with like you could use external sources, but ideally just even from your lab manual. Or just compare the that's just a discussion. Yes. Like with the trace. And then you just want us to yes. For each one. Yeah, I was good. Because I took that's okay. So sorry. So can you draw and then draw and then instead of like discussion. The discussion will be a part of this. Like the concluding discussion can be merged. Okay. They can be together. Like the conclusion could be at the very end, but I really want to make sure that your results trigger with the captions. Like a figure of this. Yeah. For each one. For each one. And so like tiny. They don't have to be tiny, but you can write them. You could draw them. Artificially like one's here, one here. Yes. Okay. That should be okay. Thank you so much. Yes, you can. This is also posted. You should have access. No. They're also on the. Oh, you took the one from the PowerPoint slides are different from the manual. And the PowerPoints are also. Yes. Okay. Thank you. No problem. No problem. Okay. Everyone's done? Is everyone done? Making sure their microscope is also on. Make sure that you're taking out the wires connect your microscope on either side. Make sure that they're off, that there are no marks or things on it. There. Take care. Yeah. Or do that's
It's just the questions are all on the slides and the line, right?
on the slides, on the manual, and your reflections from like yes. I'm just gonna say it one more time. Post-lab material includes 2 sections. Your post-lab from this week, pre-lab for next week. Pre-lab includes intro, flow charts, visuals. If you haven't shown me your microscope or diagram, please do so for lab 1. As well for post-lab, the discussion, the results of the figures that we talked about, your table for our calibration, and also the discussion questions from the manual and PowerPoint, and your pretty much reflections from today's lab. What observations you've had and how did they compare with what you expected? That's okay. You can see you weren't able to, but we'll likely do it next week. If we can, it's.
So this table is already done. Is this the table you're talking about that was we were just going through
Yes,
right now?
that's it. Nope. Also, if you could show me your post lab next week with like the table results, that'd be greatly appreciated. Thanks, everyone.
Yeah, I literally like earlier.
Yes, you did nice. Really nice. Good job. Good job.
And then I just want to clarify this thing. So the only value essentially need to know is where obviously the ocular units and where the stage meter ends up lining up to. Once it's already at 0, and then you just take that number, and then you also take the microns by like there's 10 lines and times 10, and then that's all you need.
Pretty much, and then you divide the 2, right?
Yeah.
Like 100 by 15.2, and then that's
Yeah.
your value. Try to do that for each guy though.
Yeah.
So
So essentially now, like if there a way to do it like without actually looking at it for the different?
technically you could by now. It's a bit difficult, but
Yeah.
for timing, if you've already done it for one, then it's
Yeah.
okay. So,
Okay.
maybe next week if we have the ocular micrometers, we can use it, or we'll make mention of them on the board.
There's also some values still that you could just use that to.
Those are sample ones, but the idea is if you can actually do, and if you can remind me, this would be great. We'll try to get all of them, everyone to mention what they were, like what they got.
Oh, okay.
So that way we can all fill them in case you need them for future use.
Yeah.
It could be okay.
Okay, that makes sense. All right, thank you.
No,
Yeah, so the example is one of these little birds, right? So like kind of like whatever. So all we're doing is multiplying with our 6.6.
give me 1 minute. That's okay. Right. Yeah. Okay. Yes. Good question.
Yeah, so let's see the bacteria sample or like the thing like so between a C0 and this line here. So it would be one ocular unit, right?
That's right.
So essentially, what I have to do because I have this calibration unit, I just get that 1 multiplies 6.6.
Goes on the wall.